Hey everyone, my name is Devika and I'm presenting asynchronously from Toronto, Canada. My project is on the changes in intrinsic excitability of hippocampal pyramidal neurons in an Alzheimer's disease model. 65 seconds. Every 65 seconds, someone in the United States of America is diagnosed with Alzheimer's disease. 1 in 3. 1 in 3 seniors die with Alzheimer's or any other kind of dementia. To put this into perspective, Alzheimer's disease kills more people every year than breast cancer, prostate cancer, and essential hypertension combined. The severity and sheer scope in which this deadly neurodegenerative disease affects millions of people worldwide is the reason why I wanted to delve into research regarding this field. First of all, a little background on Alzheimer's disease. When you think about the pathology of this disease, two words pop up: beta amyloid plaques and toe tangles. To explain these terms in simple terms, think of traffic jams and yarn. Picture beta amyloid as sticky gunk accumulating between roads, creating traffic jams. These beta amyloid proteins clump together to form plaques, like stubborn traffic blocks in the brain. These traffic jams hinder the smooth flow of signals between brain cells, making it challenging for them to communicate effectively. Now think of tau proteins as workers inside the brain cells, helping to keep things organized. In Alzheimer's, these tau proteins become twisted and tangled, like messy balls of yarn. This mess disrupts the normal work of brain cells, hindering their ability to transport nutrients and important molecules. Just like a tangled ball of yarn makes it harder to knit smoothly, tau tangles make it difficult for brain cells to function properly, eventually leading to their breakdown. Overall, beta amyloid plaques and tau tangles affect synaptic transmission, which essentially is the way that neurons communicate with each other. Neurons communicate over a tiny gap called the synapse through chemical messages called neurotransmitters. The cognitive decline and other symptoms seen in Alzheimer's disease are due to obstruction of these signals. However, instead of looking at the synaptic excitability, which is how responsive neurons are to signals emitted by other neurons, why not look at the actual neurons itself's ability to generate these signals in response to stimuli, regardless of the signals of other neurons? This is called intrinsic excitability. Intrinsic excitability is influenced by factors such as ion channels, which control the flow of ions across the neuron's membrane, which we will be exploring today. Without further ado, let me move on to my project. In my project, my mentor Arish Dow provided me with datasets of neural recordings from two sets of mice, a group with Alzheimer's and a control group without, from his lab in U Chicago. From this data, I began to extract features of neurons from their firing patterns. The chart I will show you momentarily are a result of combining the continuous wave-like firing patterns into a singular wave for comparison purposes. These waves are action potentials, which are integral components of how neurons communicate. When a neuron receives a stimulus, it can lead to a temporary change in the membrane potential, which is a difference in electrical charge between the inside and the outside of the cell. If the change reaches a certain threshold level, ion channels such as the sodium channel open up and make the cell more positive than its resting negative state. This triggers an action potential. Like a wave, once this action potential reaches its highest peak, it goes all the way down, or repolarizes, to reach its original negative state. During this period, the repolarization may make the cell more negative than it was before, causing it to be less excitable for a period of time and unable to generate another action potential. The following graphs are essentially a bunch of action potential waves from a neuron stacked up one on top of each other. The red dots represent the threshold value, the green dots are the highest peaks, and the blue dots are the minimum HP values, or the lowest values in millivolts of the neuron that it reaches in its repolarization stage. These three values, along with the width of each spike, the time to HP, and the amplitude distance between the threshold and the highest peak, were recorded. To extract these features, I used Python and the Spider software. It was a really great learning experience as I had been an amateur coder at the beginning of this project. Here are a sample of just two of the many figures that I've made. Overall, the results were that the neurons in the mice with Alzheimer's were definitely more excitable, with a lower threshold value than that of the control group. The Alzheimer's mice are much more likely to fire action potentials rapidly with shorter duration at a more frequent rate, which is corroborated by the significantly smaller spike width and amplitude of the Alzheimer's mice, followed by the amount of spikes, which is comparatively greater in the Alzheimer's mice than in the control. It is amazing to think that just by the opening and closing of ion channels, the way in which neurons transmit messages can be affected or altered. These findings can be used to back up pharmacological testing regarding what specific ion channels out of the 100+ types in the body are affecting Alzheimer's disease, leading to more effective and precise therapeutic interventions to modulate neuronal activity and potentially slow disease progression. Thank you for listening.